So thanks everybody for coming to this last colloquium of the academic year, or of this academic year and this semester. The colloquium series will start again in August, I assume. And it's a grand finale because we have a distinguished speaker here. He said I shouldn't embarrass him, but I'll do it anyway. So Titus Brown has one thing in common with me. He has a first name that starts with C and that he uses only as an initial. So if we want to trade secrets, you can tell me what your real first name is and I'll tell you what mine is. Yeah, no, not in public. So Titus is actually the son of a well-known physicist. So he comes from a scientific tradition. His father was Jerry Brown with a G, not the governor of California, who was actually quite well-known high-energy physicist and astrophysicist. But his son has now made good for himself also. He was trained initially at Reed College in Portland, got his PhD at Caltech, did a postdoc there, then moved as an assistant professor to Michigan State, and is just in the process of moving again now as a tenured associate professor at UC Davis. And interestingly, his appointment is in the veterinary school, even though he is really a computational biologist. And I think that this speaks to his ability, first of all, his ability to kind of go across disciplines and to get engaged in many different ways in science, but also to his strong implication in trying to teach and transmit know-how and knowledge to students. And I think for once we've seen there a veterinary school that feels that the type of knowledge that Titus can transmit is something that's going to be essential to their students. So I'll stop blabbing, and Titus, the floor is all yours.
Thank you. Microphone on? Okay. So thank you for that introduction. I never really know what people are going to say anymore. So it's always, I was watching him like a hawk, ready to stop him. Yes, so it's great to be talking here. I've been a big fan of NCSA ever since the browser. It is, I was thinking just this morning that it's very strange to be talking as a professor in a school of veterinary medicine at a supercomputing center, because I would not have, 2 years ago, I would not have made that connection. So a couple quick notes in advance. One is, this talk's on SlideShare. If you're interested in looking at the slides, just Google my name in SlideShare. Please feel free to do whatever you want with the slides. And you also should feel free to live tweet me, including anything embarrassing that I say. I'm used to it. And you can already see some pushback. I posted the slides this morning, and already people have been telling me why they're wrong. So this is an excellent example of how to crowdsource information. So when I got the invitation to give the talk, I tried to figure out what I would say. And I decided, in November, I had an argument on the internet. This actually happens to me with great frequency because I blog and I tweet. And sometimes I blog intentionally controversial things, and other times I blog things that I don't think are controversial, and then I get told they're controversial in no uncertain terms. And you can actually see over the last week on my blog exactly such a conversation. But anyway, so I thought that I would give a talk about this argument that I had on the internet. I think every talk should start with an XKCD slide. So the argument was about a guy named Uri Laserson, who I believe works for Cloudera. And he'd written a blog post that had appeared on BioStars, or he'd presented some talk slides that appeared on BioStars, which is sort of the bioinformatics discussion forum where you talk about bioinformatics data analysis and technical terms. And I don't know how many of you are familiar with internet smackdowns, but the statement that I think his take could be a bit more nuanced is the equivalent of sort of calling him out for a duel on the battlefield. So we actually had, I think, 20 or 30 tweets following on that. And essentially what Uri was saying is that the genomics community is making a big mistake by not paying more attention to what the big data community, which is to say Silicon Valley, which is where he is based, is doing with respect to big data. And we should really stop reinventing the wheel, and we should go pay attention to what Google and Amazon and so on are doing and make use of their technologies. And that's what, so his main response was, main point is lots of great tech starting in 2003 with Google File System and MapReduce. Why would you need anything else? And my response was, I don't think that will work. So it has become obligatory in big data talks in biology to point out that we're generating a lot of sequence data, and the rate at which the sequence data is being generated is outpacing Moore's law. And this has some interesting implications for people that rely on hardware to scale their analysis. And I think those implications are probably pretty obvious to many of the people in the audience. But I'll just say that this trend line really shows no sign of not continuing in the sense that sequencing continues to drop faster than whatever particular component of computation you want to analyze is scaling. So I should say the slide it's a logarithmic y scale, which is a y-axis, which is the cost per genome, and then the x-axis is year, and you can see this is right around, this is when I started my faculty position. And in fact, this dip from the Illumina GA2 sequencer is why I got involved in what I do now, because I showed up at MSU, and I was planning to do something completely different. I actually have my research proposal for my assistant professor position is on my website. You can go read it and laugh at how I didn't do any of that. Instead, what I did was I focused on sequencing data because of the gap between our ability to generate the data and our ability to analyze it. So there's sort of three I'm going to manufacture three perspectives, and everybody's perspective is actually quite a bit more nuanced than this, but well, except for the physicists. The physicists don't believe in nuance, and this actually more or less comes from George Jargovsky, who I know a number of people in the audience are familiar with, and I can point you at the paper that says this. And the physicists basically said, look, we've been generating big data for ages, and we know how to do it. Just pay attention to what we're doing and do what we do, and you'll be fine, right? And I should say I grew up with physicists. I sort of have some sense of how to calibrate the strength of their tone versus how seriously to take them. And it's a good point, actually. It's a very good point. The second is listen to the Silicon Valley folk. And Silicon Valley folk have sort of a physics level of arrogance, but in a slightly different direction. And they basically say, look, like we've developed all this software, and Hadoop and MapReduce and other things should just work for you. And like, I don't know why you're building new software. And that was more or less Uri Laserson's argument. And then the third is what we've actually done, which is ignore all previous information on how to deal with big data and instead developed our own software, which is clearly the wrong way to go, maybe. So to sort of step back a little bit and introduce the central problem that we're starting to work on in my lab, shotgun sequencing is how all of this data is being generated. At some point, it became clear to biologists, and I think Gene Myers and Craig Venter were two of the main people that realized this, that the best way to scale our ability to gather sequencing data from genomes was to, there's a lot sort of more to this, but I'll just sort of summarize it by saying if we can randomly sample and not worry about the details of what we're sampling, and we can just randomly sample many things, then we can put it all back together computationally. We don't need to do very careful sequencing of one thing at a time. We don't need to read a book one page at a time. We can feed the book into a paper shredder, and then if we feed enough books into the paper shredder, enough copies of a book into a paper shredder, we could reassemble the book from the shreds very easily computationally, even when reading the book would be one page at a time would be very slow given the biochemical techniques we had at the time. So you end up with a sentence like this that's fragmented into many different shorter sentences. And I don't know how many of people in the back can see this, but there's errors in the sentences. These are, I call them errors in digitization. We're digitizing DNA sequence, and in the process of doing the sequencing, we generate errors in the result. And the question that I want to address today, the question about the bet, which is this isn't actually something I particularly work on yet, is if we have a specific edition of a book and we sequence another edition of that book, can we, in the face of all of these errors, can we figure out what all the differences are between these two editions of the books. And this is really important because what we're trying to do is understand the genetic differences between humans, for example, or between animals or between microbes. And if we have a reference genome, if we know roughly what the genome of a white male human is, then we can go look at a wider variety of humans and we can sort of assess genetic differences between those other humans and our reference genome, and we can use that we can tie that to what I would call metadata about diseases and susceptibility and abilities and so on at a genetic level. So this turns out to be a really big, I think it's fair to say this is a really big problem moving forward, a really challenging problem moving forward. The first is there's actually a lot of data, or a lot of information, I should have changed that, a lot of information in each genome. Each human genome contains about 6 billion bases of DNA, 6 gigabases, 2 haploid genomes at 3 gigabases each. And if you cover the entire genome using random sampling, that if you reach into the genome many individual times, you need about 150 billion bases of sequencing because you need to sample on average deeply so that your minimum sampling is high. So that's 150 gigabases of sequencing, which you can compress in various ways, but basically comes down to a number of gigabytes of data, no matter what you do. There also turn out to be many, many different editions. So I've been told online that this is not a useful problem, but I'm going to present it to you anyway. If you want to look at 1,000 individual tumor cells and build an evolutionary history of the tumor in an attempt to figure out what the originating mutation was, the explanation here, I should say a little bit of biology, is that once uncontrolled proliferation starts, mutagenesis rates rise. So you tend to get cells that originate from that original tumor cell with many, many different errors in them. And you can actually do a phylogenetic analysis where you can look at all the additions at time t and backfigure to potentially what the original mutation was at t equals 0 that triggered the uncontrolled proliferation, and that has implications for treatment. If you want to do this and you want to sequence 1,000 individual tumor cells, which is still sort of on the edge of technical capability, you're really ending up in the neighborhood of around 150 terabases of data. And now we're really starting to talk about a fairly large volume of data. And I should say there's a lot of problems like this in biology. Microbial sequencing, which is what I do, also confronts similar scale of data. The other problem is that we actually have a lot of sequences, and this is where I think it's really useful to draw a distinction between what physicists do and what we're doing. In physics, you tend to have a couple data gathering instruments, at least in particle physics, and then you have a lot of people analyzing that data. Here we have a lot of instruments generating data from many different experiments, many different observations, and you have relatively little in the way of analysis capability, human analysis capability. So you may have 10,000 sequencers, and I estimated yesterday, I just made this number up, somewhere between 500 and 1,000 bioinformaticians that sort of have any idea what they're doing. And so you have a mismatch between the scale of the data being generated and the number of people that can analyze it. You have many different research groups, each with their own questions that require different kinds of custom data analysis. And then it turns out to actually be really cheap. So it's in the neighborhood of $10,000 to $20,000 for a 100 gigabase data set at the moment. And
can you just define that in terms of bytes? What gigabase?
So 100 gigabases, so I probably could. There's two things to keep in mind. One is if it's just DNA, 100 gigabases of DNA is 100 billion things that can be represented in 2 bits each. So that's, I can't do math anymore, but that's what, divide by 4. I think, and so 25 gigabytes. But you also have quality information attached to each of those base calls that comes from the digitization process. And that is much less compressible because it's very noisy. And so you end up with, I don't know, Chris, what's the, yeah, so I think we're probably talking here 20 gigabytes of data at a minimum for a 100 gigabase data set. But that turns out to be a red herring anyway, which I'll talk about later. Disk space is actually surprisingly cheap as long as you don't care if it works particularly well. Okay, so coming back to resequencing analysis, we have some addition of a genome and we want to know what all the differences are. And we have 2 types of differences, helpfully color-coded here as red for errors from the digitization process and blue for things that are systematic differences in the genome that we're sequencing from the reference genome. And we're interested in the blue and we're not interested in the red. And of course, the problem with the red is that they're not color-coded in the DNA sequence that we get out. We have to figure that out by looking at the reference sequence. So the current approach is sort of how you would do it if you were building this from scratch. You would say, okay, we're going to take all of these sequences that we get out, and we're going to locate those reads in the reference in some way that's a little bit robust to errors, and we call that mapping. And that takes one pass. And then we do variant detection after the mapping, where we take all of the reads, which are here, these sort of strings of characters, we line them up against the genome, and we count all the differences. And what we would be looking for, there aren't any true variants here, but what we'd be looking for is a line of red at one particular location. We're randomly sequencing, and we see the same base difference at a particular location, and that's going to be our variant. And this takes another 3 passes to do for a variety of reasons. And so the overall process that we currently use, if you had cancer and the Broad sequenced your genome, to sequence the cancer genome to sort of figure out to figure out what type of cancer you had, they would do something like this. They would map the reads to the genome. They'd convert that to a binary format. They'd sort that binary format, and then they'd pile up that format against the reference genome and call variants. And then it turns out that there's actually some tricky bits where there's repetitive regions in the genome, and they realign those reads against those tricky bits and sort of redo the whole thing. And it's worth pointing out this is 1, 2, 3, 4, roughly 4 passes across these large data sets. So the pros of this is that this is a really modular and flexible approach. There's actually multiple things you want to do at each stage. We're just talking about resequencing, but there are many other things you might want to do with this data where different stages along here you could do that. So there's a lot more arrows in there. The current system is open source. It's well supported, and it's pretty mature. And I have to say, as much as I complain about bioinformatics software, the current bioinformatics software approach is rather robust. And some of it parallelizes pretty easily. So in particular, the mapping of reads can be done in a massively parallel way, and everything else can be done in a somewhat more parallel way. The cons are that you're doing 4 passes across a fairly large data set, and it's very I/O intensive. And this means that it's actually unsuitable for a lot of the current cloud implementations. They're improving with respect to I/O, but it's not really a situation where, at least in the Amazon cloud, you really don't want to be writing large data files to and from disks very much. So just to give you some numbers, so again, 1,000 single cells from a tumor is 150 terabases of data. You can do this sequencing in about 3 weeks with a HiSeq X10. The variant calling requires about 2,000 CPU weeks. So given 2,000 computers, you can pretty much do this full analysis in a month. And then the only real caveat is that you then need to multiply this by all the things we're interested in sequencing, which turns out to be a lot, because there's 6 billion humans on the planet. We all, hopefully not all of us, but some of us will have cancer. Others will have, everybody's immune system repertoire is diversified, and there's lots of other things you want to sequence. So this is really multiply this problem by by 1 billion at least in the next 5 years. How do we deal with this? And by the way, we're not just sequencing humans. We're sequencing cows and dogs and cats and microbes and everything else we can get our hands on. It's just a lot of the sequencing is going to human. So where I come down on this slide is be aware of what the physicists in the Silicon Valley folk are doing, but fundamentally we're dealing with a type of data where we need our own custom approaches, and that's what I'm going to talk about next. And I think it's worth pointing out that I am more than happy to steal ideas from Silicon Valley and physicists as needed. That's how scientific progress works. And, but I think that we do need to adapt the approaches. And so that's what I'm going to be talking about going forward. OK, so going back to the internet argument, it ended with a bet. And this is actually a bet that's registered on longbets.org. And the bet that I would like to have made, I made a slightly different one because I had to leave for travel and I didn't get a chance to finish the Twitter argument. But my bet would be in 2 years, my then 9-year-old daughter will be able to analyze a full human genome sequence on her desktop computer. And you can actually follow the Twitter argument there. This is clearly not, and I meant desktop computer. It's not like she can fly over here and use your supercomputer to do the analysis. She has to be able to do it on the computer she has that she plays Minecraft on or whatever it is, right? She is my daughter, so desktop computer will probably be pretty good, but it's still going to be a commodity computer. OK, and I would like to point out, I know Uri, Frank Nothaft stepped in and had some discussions, and then my software engineer, Michael Crusoe, stepped in and tried to inflame the argument some more. And what we ended up with is the bet hinges on a nice bottle of scotch. We're talking like a $100 bottle of scotch. And I'll be out on the West Coast, so we can just drive somewhere and have a nice bottle of scotch. So it's all in good fun. This is not really a bitter argument. Although I was a little surprised at how quickly this escalated at the bottom. So this is my response to the escalation, which is I'm going to come to a supercomputing center and talk about it. So, okay. So it was nice that Victor talked a little bit about my father, who is a physicist, who passed away a year ago, 2 years ago, sorry. He had a maxim, which is never compete unless you have an unfair advantage, which is a very good maxim for scientists. So I'm cheating in at least 2 different ways. It turns out 3 different ways. So one is, I don't know if you know this, scientific genius skips generations. So my father was pretty good. And my daughter is his granddaughter. So it's not that she's my daughter, it's that she's my father's granddaughter, and she's awesome. Here is her trying to convince me of something. By age 6, she was engaging in logical, rhetorical arguments and convincing us of things against our will, which is for those of you that don't have children, it's a very scary moment. The second thing is I've been working on this for a while. We already roughly know how to do this. There's some assembly required. That's a little bit of a joke because we've been working on assembly, but we have a lot of ideas about how to do this. And then the third thing, when I was preparing this talk yesterday morning, it turns out that my third way to cheat is that a lot of other people are working on this problem, and there's a guy named Hung Lee who actually implemented most of our current variant calling pipeline, and he just came out with something that basically does what I've just described. It's open source software, and there's a preprint. So I think I'm going to win no matter what. I don't have to do a thing because somebody else is doing the work for me, but I'm going to tell you how we're going to do it, maybe slightly better than Hung Lee or what we're working on. I haven't had a chance to digest his paper. Plus, I think me coming here and giving a talk about somebody else's work would be slightly strange, although I should try it sometime. So the remainder of the talk is to point out that really what we have here is a confusion of data versus information. I'm going to talk about some of our streaming approaches to lossy compression that we've used together with compressible graphs that we've used for soil metagenomics. I won't talk about soil metagenomics. And then I'll describe a little bit about sequencing errors and variants using graphs. So when you look at how we load this data in and analyze it, one of the common approaches is you load all the data into a graph. And if it's a chapter, if it's a book, it's a connected sequence graph of sentences, essentially. And one of the things that started to trouble me, is a big problem in this area, is that you run into 2 different kinds of scaling behavior. One is if you're sequencing a human genome, as you generate more and more data, the amount of sort of true information in that data set levels off because there's a finite amount of information in the human genome, and you sequence more and more, and eventually you've seen all that information. And then there's another set of another component to data, which is the erroneous data. And errors are linear. Every time you do digitization, you get a different error. You get different errors, and the error rate's more or less constant with the amount of data you generate. And so essentially your error rates simply climb as you generate more data, which means that your total size of your graph keeps on climbing as you sequence more deeply. And this is because of the random sampling and behavior of errors issue. And the reason these sequence graphs scale so badly is basically what you're doing is you have to represent all of the real variation plus all of the errors in that one graph. And so here, again, I've helpfully highlighted the errors in red. And you can see that what we're doing is we're lining up all these sequences against each other, but then we have to represent the differences. And the differences simply accumulate more and more and more as you sequence. What this means in practical terms, and this is an old graph, I should put a date on this. This is from like 2009, 2010, when we first started melting down computers. We took an 80 million read data set from soil metagenomes. So that's 800 megabyte, no, 8 gigabyte, there we are, 8 gigabyte data set, I think. 80 million reads that are 100 bases long. And we just ran different subsets on a computer that had 125 gigabytes of memory. And what you can see is that the memory usage of these assemblers is more or less linear with the data set size. And for those of you that are computationally expert, the pain point starts around 100 gigabytes of RAM because it's a little bit hard to find computers with that much memory sort of casually. And I should point out that the X scale here can go a factor of 30 out to the right, which is where you start to cause problems. We estimated that for this full data set, which was 3 billion bases, we would need 3 terabytes of memory in order to just run the assembler on it. Now, there are many things you can say about this situation, one of which is like, well, don't you need better software? And my answer to that would be yes, and that's what we've worked on for the last 5 years, so exactly. But this is actually a characteristic scaling behavior of the graph itself. So if you have any linear algorithm, any algorithm that's linear in memory, storage with respect to the data set, you're going to have this kind of scaling behavior. The slope may be different, but you're still going to have this scaling behavior. And that's what we need to change. What we want is we want a behavior that changes, that scales with the blue edges, with the amount of information, the amount of true information in the data set. So a key concept in sequence analysis is coverage. If you have some unknown genome or known genome and you sample a bunch of data from that genome in an experiment, the coverage is defined as the average number of reads that intersect any given genome, any given location in the genome. So here, any point you draw down from the genome will intersect about 10 different reads. And so the coverage here is approximately 10 because you intersect 10 reads as you go down. And the coverage distribution basically follows a nice Poisson distribution. You're reaching in randomly into a genome and pulling out reads. And if you graph the number of times you see each base, and each base is covered by a read, you see that you can define the sort of average coverage, which is here on the right, 10. This is a synthetic, a simulated data set. But you get results that look pretty much like this from real data. And you typically need in the neighborhood of 10 to 100x coverage. So the median, the average of that curve needs to be between 10 and 100 to be at the point where you've observed every base enough times to accurately call that base without too much fear of error. I don't know if that makes too much sense. But to reiterate, basically, if you only see a base 0 times, you can't call that base at all. If you see it only once or twice, you don't know if it's going to be an error or if it's going to be real. If you see it 10 times, you're good. You can pretty much call that base accurately off of the data. There are a lot of details in here, but that's how the statistics work out. And the main observation we made was that once you get past a number of times, like 10, that you've seen a base, you don't actually need any more data. That's not to say you don't have to collect the data. You have to collect the data because you're randomly sampling from the population. But that data is unnecessary for the perspective of computational analysis. So we developed an approach called digital normalization that walks through the data set, piles up the reads against a reference graph, so not the true sequence, but against a graph of the reads that we're building in an online manner as we walk through the data set. And then as we pile up the reads, we ask how many times have we seen this region of the graph before? And at some point we say, you know what? We don't need more information for this locus in the graph, so we're going to stop collecting data for that locus in the graph. So what you end up with at the end is you end up with a bunch of reads that correspond to information up to a certain coverage level, plus a bunch of redundant reads that aren't needed for later analysis. And this has a very salutary effect. So you can go for some real data sets, you can go from like an E. coli data set with 31 million reads and eliminate all but 600,000, which represent the novel information, which represent a complete E. coli genome at a coverage of about 5x. The assembly time goes from 1,000 seconds to 63 seconds, and the memory goes from 11 gigabytes to half a gigabyte. The entire process actually doesn't take very long, and it's surprising. It's still surprising to me 4 years later that it works. The real point here is this is the scale of redundancy of sequencing data. We're really over-sequencing genomes. A lot of what we're sequencing is errors, and if we sub-select the reads, we also sub-select the set of errors we have to store in memory, and we get rid of the rest of the errors. So this is essentially an error correction approach where we're choosing a subsample of the data set and eliminating the vast majority of errors. And this lets us do a surprising array of analysis directly on commodity computers. We actually use it as a pre-filter, so we take the data that comes out of this and feed it into a regular assembler. We didn't want to write our own assembler. Other people have done great jobs of that. So what we're doing instead is cherry-picking a set of data that we can feed into downstream analysis approaches. It's also, interestingly enough, nearly perfect from an information theoretic perspective. And this is going to be in slightly odd units, but if you take a simulated genome with 400,000 words in it, and you generate 8 million of these unique words in the sequencing data set, most of those unique words come from errors in the digitization, and then you run it through this normalization process, you lose 2 real words, and you eliminate 3 million erroneous words. And you keep only 20% of the data, and you can actually do multiple passes on this and do some error correction, some other things. And essentially what we can do is we can put informational bounds on how much we're losing in terms of real information, for what opportunity in terms of how much false data we're rejecting. And I usually have some slides in here about JPEGs and lossy compression and how we can tune this so we can either lose a lot of information and keep basically only the basic structure of the assembly graph, or we can keep the full assembly graph while retaining a lot more data. At the end of the day, what we've done with this is change the way analyses scale. So we've changed assembly in particular, where we're reconstructing the text from the raw reads, from scaling like the total edges here to scale like the true edges here. So this is effectively streaming lossy compression. The 3-line algorithm that we actually have implemented that actually does this is looks like this. We look at each read, we estimate its coverage, which is a very simple operation based on k-mers. And then if it's still an interesting read below some cutoff, we pass that read on. So this is a generator in Python. And it's important to note this is literally a 3-line algorithm. It just took us 4 years to figure out which 3 lines to put, right? And I think that this is something that I've seen a couple times now in my sequence analysis career, which is that if you can often boil a lot of these problems down to a really, really simple underlying concept that can be implemented in a very simple way on top of a robust library. And for whatever reason, we haven't done that so much in bioinformatics, and that's a direction that I'll tell you a little bit more about that. The other really nice thing about this is that if you want to try and make this a more generic statement, digital normalization can detect this information saturation in a stream. So as the data comes in, we can assess whether or not at this point in the stream, we've saturated information at a particular locus within the graph, particular locus in the genome. So here you can see for RNA, for transcripts where there may be high abundance transcripts and low abundance transcripts because of biology, basically, we can say, we have enough information to assess the sequence and structure of transcript 1, we just haven't collected it enough for transcript 2 in this stream. And what this lets us do, and this is a new paper that was brutally rejected from PureJCS and that we're revising, we've been able to show that this generically permits us to take a large collection of analytical approaches for looking at sequences and move them into what's called a semi-streaming approach. And semi-streaming means you actually have to go back and look at some of the data twice, because in your first pass through the data, there's a lot of novelty, and you actually have to go back and look at that novelty again in order to capture all the novelty in the data set. So the first pass goes across all the data, and the second pass looks at a subset of the data. And what this ends up being, it's like a 1.2 pass sublinear memory analysis of genomic data sets. And if you have no errors, you get the dashed line for an E. coli data set. So you walk through like 5 million reads of data. And by the time you're 20% of the way through the data set, you've essentially seen all the information. If you have errors in your data set, you keep on collecting novelty, because you can't tell, with this approach, you can't actually tell the difference between errors and, with this very simple approach, you can't tell the difference between errors and true novelty. So you have to collect, you take a conservative approach and collect some more errors just to make sure you're capturing all the novelty. But this is actually real, the blue line is real data. So it actually works quite well. This also turns out to be a real simple algorithm. It's essentially, you add a couple lines into the first pass, and then you have the second pass is the same thing as the digital normalization. So, and we actually claim that you can add a whole variety of k-mer frequency approaches, k-mer spectral analyses into this framework, including error correction and variant calling directly into this loop by including them in the streaming analysis. And so this gives us error detection, and it turns out if you can tell what an error is, you can also tell what a true variant is. And so we can actually line up all these reads and just look down the column and say, gosh, we see a lot of C's and we see a lot of A's here. That's what's truly at this position. And so in the end, what we're working towards is sort of a single pass, reference-free, tunable streaming online variant calling, which is very different from that 4 or 5 pass approach I showed you earlier. In practice, what we've actually been doing is building these little components. So we've been trying to build a general framework for real-time and streaming data analysis, and we actually are getting close to the point where we can take the data in real time off the sequencer and analyze it as it's coming. That's actually really useful for the clinic, where latency starts to matter. Some of our software is already being used by Illumina, which is the main sequencing platform. And there are other sequencers coming along where you can actually feed back into the sequencer. You can say, I know you're looking at this molecule. Let me tell you that molecule is no longer interesting. Kick it off the sequencer and load a new one in, this with a nanopore technology. So we're thinking about how to do this. What we're trying to do is not actually interact in an engineering sense with the sequencers, but build the underlying theory and implementation that lets them swipe our ideas and use them for fun and profit. And so We've now implemented, this code actually works. We've actually implemented several things in a completely semi-streaming approach via generators in Python. So the read enters, goes through a set of components in real time, and then comes out the other end in real time. And we can string these together in a variety of different ways. And it actually permits a pretty wide array. Like my sense, my intuition is that we can actually do all of primary sequence analysis in this semi-streaming manner, which would be quite nice, especially if you view these boxes as being potentially on different machines. So you can have different, you can have high performance I/O machines versus big memory machines versus machines with big disks, and optimize your cluster configuration in that way. So we've implemented, we have some other stuff that connects then to variant calling, and we can actually look at an E. coli genome in about 5 minutes from scratch using a, I think it's a couple hundred megabyte or couple gigabyte data set in about 40 megabytes of RAM with a single thread with no optimization, primarily in Python. We can do a full variant calling analysis on E. coli. We believe that scaling to humans should be feasible, and we have 18 more months before I lose the bet. So my guess is that what we're really most interested in is not actually providing that implementation. Our experience in bioinformatics is nobody's going to use our code anyway. What they'll use is they'll use the ideas, and they'll re-implement them themselves. So that's why we're fixated on this idea that if we can theoretically understand things and implement them in a very simple way, other people can steal those ideas much more efficiently than if they have to buy into our body of code.
You have some quality metrics associated with your bet, meaning how many false positives and how many false negatives are you allowed?
So you have to, so the question was, are there any quality metrics associated with the bet? And I'd like to bring up I have two responses. One is variant calling itself is actually quite, black art's a interesting way to put it. I think that I would say that variant calling right now is surprisingly variable. And I think that when you realize that the failure mode of the bet is that we both buy bottles of scotch and give them to each other, I feel like that's an okay failure mode. So I'm not too motivated to come up with really hard constraints. But I mean, it's a really good point. Where I really want to go with this is to get to the point where we have parameters that we can tune, and we can encompass either very liberal variant calling or very conservative variant calling. And all of this matters, as you know, for the biology that you want to do with it. Sometimes you don't care if you catch everything because you're looking at long-range effects. Other times you care deeply about a particular thing. You don't mind about the false positive rate. So we want to provide that range of approaches rather than settling on one specific one. Okay, but so getting rid of all the bet stuff, the real point is that what we need in all spaces of science, but particularly in sequence data manipulation and biology, we need these sort of well-founded, flexible, algorithmically efficient, and also high-performance components for sequence data manipulation. We are building some of these on a streaming and low memory paradigm, which if we can make it work, I think is a really good direction. And then we're also building out a scripting library so that you can compose these operations easily in practice. And so we're trying to encompass this whole space of building novel theoretical approaches, algorithms, and data structures, and then building them all the way out to actually doing effective sequence analysis with them. And I think I should just do a shout out. I mean, this is very hard to do, as I think many people in the audience know. And this is the kind of thing that that I think the future of much of science depends on, is actually having well understood operations that are implemented well for data analysis. And so I'm very passionate about trying to figure out how to do all of that at once, which is not sane. So to get back to the title, should we scale compute or algorithms? I feel that the Silicon Valley approach of take a fairly boring problem and throw lots of computers at it has its limitations. You can certainly solve a lot of these computational biology problems by scaling to many computers. My question is how feasible that is moving forward as a field. I know there are many problems that require big computers and many processors. Based on information content alone, I think it's very clear that genome sequencing should not be one of them. And given the scale of the need, I would argue that that's actually really good. We want to be at the tricorder phase, where you walk into your doctor's office, they take a sample of your blood, they get the answer, they don't need a Beowulf computer in the back room, and you can do that on a scale of millions of people a day, because that's the scale of the biomedical need. We can talk afterwards at the reception or in the questions and answers about the many other biological problems that do require big compute, a lot of the machine learning and other things and modeling. So this is not a blanket statement about biology. This is really sequence analysis should be tremendously boring, and I'm hoping to make it tremendously boring. Okay, so now I'm going to go off a little bit. Biology's gotten, the sphere of biology that I work in has gotten really distracted by these shiny toys. We can finally get data on systems that we had no access to previously. Soil and non-model animals and agricultural animals. It was always possible to get data on humans because, we're the ones funding the science. But now data is plentiful across the board, and now our challenge is, of course, returning to what it should have been in the first place, which is understanding. And it's a little hard to convey just how little we know about biology. This is actually a very conservative statement. When we go out and we sequence soil, which is actually what I do research on, or one of the things I've done research on, we typically have no knowledge whatsoever of what 50% or more of what we get off the sequencer does in any way, shape, or form. Literally, it's conserved unknowns is the best we get. Like, other people have seen it. So, and in fact, we really only have any idea what about 20% of what comes out of an environmental sequencing run does. And I have a very long blog post on what if somebody, I like to think about what would happen if somebody dumped $100 million on me, just hypothetically. Talk to me later if you are interested in dumping $100 million on me. And it's actually a really challenging biological question. How do you characterize all of these unknowns? You could have an army of graduate students, and that might be one success mode in a certain way, not for the grad students, but for you. Or you could do some other things, and there's a whole panoply. So you can create a sort of balanced portfolio of how to attack these problems. So here's a joke. I was going to give you my 5-year vision, but I don't have 20/20 eyesight. No? Okay. So it's 2015. 2015, 5 years, 2020 year. Yeah, okay. My wife asked in advance that I apologize for this joke. It came via Adrian Holavati. Anyway, looking out, one of the things when I started, I would love to pretend that I was somehow foresightful. But in reality, I describe my whole career as a high-speed random walk, slightly guided random walk. But I will say, having come from a physics lab, having done a lot of research with physicists as an undergraduate, when I started my faculty position in 2008, and I saw the output of the Illumina sequencer, and I understood the scaling properties, I knew that in 5 years we would be facing essentially an information apocalypse. And in another 5 to 10 years, that will simply have increased more. We're going to have nearly infinite data. Whatever we can get our hands on, we will be able to sequence, analyze, extract data for in a variety of ways: genomic, transcriptomic, proteomic, metabolomic. Matt Turk was just telling me today about some things they can do where they can take an image and they can actually do, I'm probably going to get this wrong, but like volumetric analyses of metabolites in a three-dimensional space in little cubes in that three-dimensional space. I mean, and you think like, okay, well, that's probably a small amount of data, right? No, right? It's immense amounts of data that we don't know how to understand, we don't know how to analyze, we don't know how to integrate. There's all these issues of data integration that biology is ill-prepared to deal with: registration, cross-validation, integration with models. Eventually, we're going to have to come back to modeling to understand and integrate all this data. And that's really the next challenge. Just to give you a different example than human biology, I'm partnering with some people at the Woods Hole Oceanographic Institute who are interested in the primary cycle and have gone up and down the Atlantic, the South Atlantic, sampling, and they've sampled a variety of different data sets. They've sampled DNA and RNA, they've sampled protein, they've sampled metabolites, and they've looked at physicochemical data and then also hydrographic data. And all of these things have different precisions, they have different sample frequency, they have different amounts of data, they have different challenges. And if we're interested in understanding the microbial dynamics of biogeochemical cycling, we need to integrate all this data together. And I don't think it's a stretch to say that we have no idea how to do this at all. Based on what the physicists do, this is a diagram that we came up with. I came up with a physicist for a training grant that also got brutally rejected. You'll sense a theme that we need to be able to go from raw data and filter data, build these metagenomes and understanding of what's present in the microbial population, turn those into metabolic networks, turn those into a community ecosystem model, and then use that to build predictions that turn into experiments. This should be fairly familiar to anybody that really thinks about how other fields do data integration, and we just have many challenges. Biology is tremendously complicated. We know very little about it, and we just need to start integrating our analyses in this feedback cycle. So what do I want to do next? So a few thoughts on sort of pragmatic and practical next steps. So I sort of have three basic thrusts. One is I want to enable scientists with better tools. The second is I want to train a bioinformatics middle class. And the third is I want to accelerate science via the network effect in open science. And these are all somewhat hand-wavy, but that's what energy is for. So a really great point, David Hausler, who works a lot on human genomes, it's fair to say, what do we do now? He said, business as usual simply cannot work. We are looking at millions to billions of human genomes in the next 5 to 10 years. Our current way of looking at genomes is to look at them one at a time. That's really not going to work going forward. And we need to change the way we think. So enabling scientists with better tools. This is sort of what I've already sort of said a couple times. We need to build these robust and flexible computational frameworks. I know I'm preaching to the choir. I think I'm preaching to the choir on this one, right? We need better tools, we need better theory, we need algorithms, and we need software implementations, and then we need to train people in how to use them. The scale of the training challenge here, which I'll get to in a second, is massive. And very, very important, I'm speaking to you, Victor, we need to stop pretending that we can develop black boxes that you can feed the data into and with no understanding will simply give you a publishable answer. He doesn't believe that. Which is what every biologist wants, right? I'm actually thinking on my Davis office door of putting a big black box and say, deposit data here, science paper will come out the bottom. And it's really a problem because there's so little training in biology at the undergraduate and graduate level on what data analysis means that the whole field has sort of is operating at a level of abstraction that's simply too high in thinking about data analysis. And this is one of my big efforts, is really in education and training. And it's going to be a situation where every biologist needs to be able to do some aspect of their own data analysis, even if it's only running software that other people have developed. I don't want most scientists developing software, right? Like, it's just a disaster. But what I do want is scientists understanding how to control the software, put controls on the software that other people have developed. And so what I'm interested in doing at Davis in particular, but also globally, is working with other people, is building this sort of cohort of data-intensive biologists who can use all of this as sort of a really unremarkable part of their research. Like, not like, oh my God, look at this data. Like, oh man, that's going to take a week. Fine, right? It's just something you do. And so at Davis, I'm scaling up training in collaboration, in partnership with software carpentry, and as a friend of data carpentry, I'm starting up a data-intensive biology training program at UC Davis that's focused on running workshops, trying to cross the divide between novice and master class, building open materials, and generally trying to be a robust partner in this sort of worldwide conversation about the training disaster in biology, the data training disaster in biology. And then here's a really generic slide about the network effect in open science that I'd like to refine in some ways, but right now too much of what we need to do in biology is locked up in labs. Methods and data is generally inaccessible within the biological literature. And I feel like if we can somehow get this sort of many-to-many system going on where I can discover data sets that are of utility to me in other people's labs, then I know to talk to them about potentially collaborating. And that should lead to a massive acceleration in progress, or just massive confusion, but those are more or less the same things. So this is the subject of a Moore Foundation grant that I got, one of the Moore Data-Driven Discovery Investigator grants that Matt Turk in the audience also has. And mine is actually for a sort of pie-in-the-sky idea that maybe we can drive data sharing via decentralized model, a distributed graph database approach. The award is, like all my single PI grants, the award is available to read on my website. Please, I'd love to talk to you about it. It's all very hand-wavy right now. The Moore Foundation gave me the money without me telling them what I was actually going to do, which is awesome. But it does mean I have to figure out what to do. So we'll see how that goes. From a larger perspective, I have what I like to call 100% buzzword compliant research vision. And these are all Silicon Valley buzzwords. I don't know if, not all of them, but most of them. So I want to enable frictionless sharing. I want to enable permissionless innovation. If you have a new data mining approach and you want to apply it across other people's data sets, you shouldn't have to ask them for permission until you want to publish it. And even then, it shouldn't be so much a matter of permission as like, well, obviously, just cite me, right? That's how it should work, and it's not how it works currently. One of the big problems with cyber infrastructure efforts right now is they plan on continued funding. And as a cyber infrastructure aware audience, I think we're all aware that continued funding for really anything is not guaranteed, but cyber infrastructure can be a particularly challenging case. And so I really want to build this in a federated way on open and cloud computing, open source and cloud computing. And then, the real trick, of course, is advancing the field, solving people's current problems while remaining agile in such a way that as new problems come along, we can tackle those. And I have no idea how to do a lot of this, but that's what tenure is for, right? So thank you very much. I've been funded by an unreasonably large number of people, including the USDA, the NIH, the NSF, and now most recently the Gordon and Betty Moore Foundation. Please feel to drop me an email with critical questions about why pointing out why I'm wrong about everything I just said, and I'd be happy to take any questions. Thank you for listening. All right, do I face the, yes, please.
Just a quick question. I know it's not a big deal when you're doing soil metagenomics, but have you looked at the effect of your pre-normalization on copy number variation and
Sure.
insertion deletion detections?
Very
That's
important.
actually a critical aspect for almost every aspect of human biology.
Very important point. So the question is, if we're doing this abundance normalization on the sequencing data, do we lose resolution on certain important features of the graph, basically, or other counts? And so the short answer is, in the cases where we've looked, we can find parameters that guarantee the retention of certain structure. What we're actually working on now is a slightly different approach that's considerably more specific, that lets us guarantee the retention of graph structure in certain ways. And then we're working to do both theoretical analyses of information retention of those things, as well as checks on real data. So the short answer is, in a lot of cases, we know that we retain the things we're after. And we have approaches that we think can retain everything that we're after, but that is a 2-year in the future guarantee. The short version is for abundance in particular, which has been a challenge. I represented digital normalization as a way to get rid of errors. So it turns out that you can keep the semi-streaming approach that we're just sent out for review gets rid of the errors in a different way. So we retain the abundance. And then our next paper will probably be on error correction, which actually fixes the errors rather than getting rid of them. And that should be even better for abundance. So we think we can replace mapping entirely that way. Large-scale graph structure, I think we're at the mercy of the fact that our sequencing approaches with the short read stuff that we're doing right now don't get those either. And so what we're interested in doing is building hybrid graphs where we have the short read data, and then we have the long read data, and we can actually label the graph. We have a lot of technology for doing this. I guess I would just say at the moment, I would love to see other people jumping into this field because we have way too much to do. And there are other people, but we really like the streaming and low memory approach, which not that many people are doing. So I totally get what you're saying. I have no idea is the broader perspective, but yeah. Victor.
Towards the end of the talk, you formulated an idea that sounds suspiciously like the NIH Data Commons framework, and actually something you may have heard about here at NCSH, the National Data Service. The whole idea of, it's okay, a very loud voice. Basically having an environment where you can find and share data.
Yeah.
And do something.
Yeah.
So are you just mouthing the same ideas as the funding agencies, or do you have a maybe more progressive and lighter way to
You're
approach
determined
this?
to lead me into a path where I will get myself into trouble. I really appreciate it. So
Nobody here works for NSF anymore.
So first of all, I talk with all of these people. So I think at least in some cases, they're mouthing some of my words. In general, but really, in general, the set of ideas has been around for a long time, and the challenge is executing, as you well know. So where I would say that I am more progressive than the NIH and the EBI is, and this is something I'm trying out, so I'm not sure how well it's going to work in practice. So I can go totally off the deep end with this. Maybe I'll go halfway off the deep end with this. So we're no longer, so all of our current data mining approaches for genomic data and all of our current repository models for data are centralized. They may be federated, but they're generally federated in such a way that they're strangely centralized. There will be a few big data centers, and the data will generally be shared between them. And maybe it'll move, maybe it won't, but there will be these big databases. And that doesn't scale in a number of very important ways. It also doesn't work legally. So I don't think there's a future where Brazil sequences all of their citizens, and we have access to all that data in an unrestricted way, right? So what I really want to enable is data mining approaches. You can think of this as a differential privacy issue as well. I want to enable data mining approaches that let us ask questions of remote data sets. It doesn't require us to have full access to them. Again, not a novel idea, but I want to do this in a fairly flexible and generic way. And what I'd really like to do, so sort of, this is where I go halfway off the deep end. What I'd really like to do is to enable a container where you can send containers around, and you can say, hey, here's a registry of containers, like Docker-style containers. Run this container against your data set of this type. Suppose you're sequencing infectious diseases in your country. Run this container in an isolated environment against that data type. It will turn green. If it turns green, we will give you this much money in order to have access to your data set. If it turns red, your data set's not of interest to us. And then, of course, this all works via a decentralized Bitcoin protocol. But what I really want to see down the road is automated contract negotiation and exploration of remote data sets in such a way as to preserve the original rights but still connect people. And we don't have that currently, and this is a totally crazy idea, and this is a totally crazy idea right now, but it's totally feasible in theory. And I think that we can know we're heading towards a future where we will not have global centralized access to any data, to the data sets we're interested in. And that's the future of biomedical data science is going to depend on figuring out ways around that. That would be my overall statement. So.
Yeah, so this crazy idea seems conducive and reminiscent of the citizen science idea in astronomy, where data are being shared across the regular run-of-the-mill individuals who are simply willing to lend their computer resources toward it. And the person doesn't need to see the data or have access to those data. Just sort of distribute the computing and distribute the data storage. And especially if data are containerized so that the user could not see the data, that would be particularly conducive.
So there's a couple different, so I read a lot of science fiction and fantasy, in case that's not clear. So there's a couple ideas that have come forward. There's the crowdsourcing idea, which is sort of have many people analyzing data intelligently, and that's one of the ways that citizen science is progressing. There's the, I've just now forgotten the distributed compute system out of Berkeley, BOINC, where you essentially have containers that you execute on your local hardware to do compute, and they send the results back, and you don't know what the result is, and you don't know anything about that. And I think that my basic statement would be exactly that. The real question is not whether this is computationally feasible. It's clearly, 6 months. If it became important, we could implement most of this in 6 months. The question is, how do we hack the scientific culture so that scientists don't stand in the way of this? Because right now, most scientists would be like, I don't know why I would do any of that. I don't know why I would share my data. It's only ready to be shared when it's ready to be published, and I will get proper credit for it. And you point out that they're standing in the way that the people are dying because of this attitude, and the response is, yes, but I have my next promotion to worry about. And this, it's how do you align the incentives? Anyway, you know the drill, right? How do you align the incentives with the correct incentives with the career incentives? And we're very bad at that in science at the moment. I think that we're missing. Every week goes by, we're missing massive opportunities to make progress. So I guess I've got tenure now, so I don't care anymore. But no, but seriously, like how to hack that's the problem, and it's a generational problem. And I'll just refer you to, it was Dirac's quote, was it? Or anyway, there's a quote about science progressing one funeral at a time. It turns out that it is technically illegal to hasten that process, but I think that data science is an interesting opportunity to turn over biology, how biology does things. This is being recorded, isn't it? Uh-oh. Sorry.
And just one last thing. I know you talked about data or code reuse. One of the things that I've noticed a lot in my own work is code reuse is hastened when people actually develop libraries that can then be plugged into other software. And I know it's something that just basically doesn't happen in biology because people don't actually know how to properly engineer their software in biology. But it's something that, like, for example, when you're just producing a Python library or a Python set of code, that means that it's locked into a specific set of tools. Whereas if you develop a library that has bindings that can be used by other languages, now you've got a core that can be used elsewhere. Have you thought about, like, especially some of these techniques sound really interesting, and I'd love to use them in my pipeline, but having to use exactly your interface is
Sure.
always going to be a problem.
So preaching to the choir, we have a C++ library that's wrapped in Python. There's no reason we couldn't develop other language wrappers. I used to have a Perl one, actually long time ago. One of the reasons I was laughing is if you go back 5 or 6 blog posts on my blog, I apparently somewhat impolitically yelled at somebody for moving away from a library model towards a standalone binary model. And I got taken out behind the woodshed on that one. So, and there were a lot of people that made the argument. I mean, there's a lot of really bad arguments made around, like, C++ should be good enough for everybody, including bench biologists. And then there was standalone binaries are the way forward because of installation issues. And then, and at some point you go, well, at some point you just get tired of arguing. I mean, apparently not me, but you get tired of, you just want to progress with your own research. So I'm trying to do this as what I'm trying to do is have a demonstration. And I think I and the others that are doing this approach are having an impact. Scikit Bio is another thing that I would highly recommend looking at. It's just going to take a while. We're training people in basic software development practices. We're trying to figure out how to make this work. The funding people are not necessarily cooperative. I'm finding it difficult to argue for maintenance of software because there always has to be a novelty component. All of the standard things. So I'm totally on board with what you say. My question is, how do we make it happen? And my current approach to making it happen is if we can scale variant calling by a factor of, say, 50,000, and nobody else does, then either they have to re-implement our software from scratch, which is probably what they'll do, frankly, or they'll have to use our software. But either way, it will be an advantage because we won't be locked into 20 years of legacy code. So that's a strategy. We'll see if it works. I have no idea.
Okay, I think
Thank you.
If you want to have further conversations with Titus, you should move to the refreshments area out here. There's some crackers and cheese and fruit, I think, and you're all welcome to partake. And thanks again very much for this.
Thank you.